Simon, for the very kind introduction. And thanks for everybody for being here. It's a great pleasure for me to be a part of this NGSE series. A lot of really stellar scientists, postdocs, and PhDs have kind of contributed to this already. So it's a great pleasure for me to be one of those people. So as Simon said, I will be talking about sort of multimodal microscopy of halide perovskites. And it's work that I've entirely done during my PhD and a little bit afterwards in the group of Samstrax in the University of Cambridge. So without further ado, I'll give you just a bit of a brief outline. I'll talk a little bit about why sort of photoluminescence is a nice and natural probe for semiconductors and for solar cells in particular. I will kind of go into a little bit of detail explaining the experimental techniques, some kind of a few tips and tricks, I suppose, how they work and things to be aware of if you were trying to do something yourself, any caveats that might catch people out. I will talk about these sort of multimodal probes that we put together, where we put together several different microscopy techniques, each with a different contrast mechanism, and kind of point them all at the same area and try to develop sort of a correlative understanding of relationships between, say, the structure and the chemistry and then the optoelectronic performance of a material. Then we'll kind of finish off at the end with sort of extending this sort of framework to the operando. What can we learn when we operate a device and point on all these different microscopy techniques to it, and then a few conclusions. So first of all, back to basic physics 101 or whatever. The sun is a black body, which is sort of a perfect absorber and emitter. And this is the sort of spectrum of the emission or the radiation of the sun from Planck. This is sort of Planck's radiation law. And it can sort of be written as the product of these two contributions. One is the photon density of states. So this is, what states are available for photons to be emitted into. And then we have the sort of occupation function, or the Bose-Einstein occupation function. What proportion of those states for a given energy are occupied. And so the product of those two things gives you your black body spectrum. And effectively, the only free parameter here is the temperature. And so it gets hotter, gets brighter, gets bluer, all that good stuff. So real materials are kind of gray bodies. And so what that means is you have to add in just a few little things. For real materials, it means that their absorption coefficient is finite rather than infinite in the case of a black body. And that means you need to just take into account the absorption spectrum here in this added term, but it's otherwise pretty much the same. So again, you have your photon density of states. You have your photon occupation function. This now has an added term, which is the quasi-Fermi level splitting or chemical potential difference, which I'll get into momentarily. And you have the absorption spectrum of your material. And so if you multiply these three things together for any sort of material, you end up with your lovely photoluminescence spectrum. And so this actually is quite nice because it means that you can measure the photoluminescence spectrum, and with sort of a careful understanding of where all these terms come from, you can actually pull back out either the absorption spectrum of your material, particularly below the band gap, or this quasi-Fermi level splitting, which is effectively a metric of the maximum voltage that your solar cell could produce. So I'll get into that in a minute. But it means that photoluminescence, if you measure it carefully, can really be a nice probe for very device-relevant metrics. So here's some graphics that I made, Hollywood style, being in California. We have our semiconductor with a valence band and a conduction band and a Fermi level in the center. In the ground state, effectively all of these states will be occupied by electrons and all of these states will be empty. We shine in our photon and we get incredibly, an electron absorbs its way up into the conduction band. And if we shine light on the steady state, we basically will have some steady state excited population of electrons up here, and we'll have depleted some states in here. And so we'll have a steady state concentration of holes. And so we can sort of write, given that all these electrons are sort of in thermal equilibrium with one another, we can write a quasi-Fermi level for that population of electrons and a quasi-Fermi level for this population of holes. The more light we shine, the more electrons get populated, the more holes get populated, and the more there's an energetic difference between these two Fermi levels. And so this means that these two populations have a different potential energy, and populations with different potentials can do work. And so this is the sort of well-known photovoltaic principle. The analogy I always like to use is that we have a nice dam with electrons at the top and holes at the bottom. So photoluminescence is a really good probe for this, and that's kind of the take-home message here. And so when you have a material that can absorb light, you inherently couple the ground and excited state with a photon. And it means that the inverse process is also possible. So you can have an electron fall back down and emit photon again. And so that's unavoidable. But then, as we know in the field, there are several other mechanisms that could also result in electrons losing their energy in a non-radiative way, where there's no emission of a photon. So this is either via a trap state in the middle of the band gap, or via an Auger process where an electron, for example, gives some extra energy to another electron. And so this is an analogy I've seen a lot in the field, where we want to basically maintain as large a quasi-Fermi level splitting as possible. It's sort of akin to trying to fill up a bucket with some water. The higher the level of the water in the bucket, the more voltage our solar cell can produce. And every additional pathway adds a hole in the bucket and reduces the sort of steady state level of the water. And so we basically, as solar cell device physicists and chemical engineers and all the rest of it, our job is to plug those holes. And so one metric that I think people in the field are familiar with, of course, is this sort of photoluminescence quantum efficiency, which basically tells you for every, say, 100 photons that you shine into your material, what fraction of them recombine radiatively. And from Ross's relation, we can basically relate this to the voltage. So we'll have some radiative limit voltage, and for every order of magnitude drop in photoluminescence quantum efficiency, we'll lose about 60 millivolts. And so this voltage is equal to the quasi-Fermi level splitting in the ideal case. And so there's your simulation. When you have an ideal solar cell versus with some non-radiative recombination, we'll see a big drop in the voltage. Bad stuff we want to avoid. So very briefly, in terms of actually extracting quasi-Fermi level splitting from photoluminescence, we have our lovely, carefully measured PL spectrum that we liked. We have our generalized Planck's law to account for the quasi-Fermi level splitting and the absorption. The sort of most common approach in the literature is generally to assume that above the band gap, the absorbance is more or less a constant and equal to 1. Just assuming that you're basically absorbing almost all of your photons above the band gap. And if you do that, you can sort of rearrange things by taking a log and having an e squared over here. And you basically plot this log of the PL divided by e squared versus energy. This is all a big constant term. And so your intercept will be related to a constant plus the quasi-Fermi level splitting. So you just take the log of your PL, fit the linear term above the band gap, and you can extract your quasi-Fermi level splitting that way. An alternative approach that was developed by Katahara and Hillhouse in the University of Washington, you can basically develop a model for the absorption and fit the entire PL spectrum. Sorry, that's a bit small, but it allows you to, in theory, pull out what you think the below band gap absorption looks like, whether it's a traditional Urbach tail or something more complex. But I hope this slide kind of gives you the idea that photoluminescence can really tell you quite a lot about a material, which is quite cool. So perovskites, I won't give you the spiel. I think it's not the audience for me to explain what a perovskite is, but there it is. We can talk about it more if you haven't seen it before. They're solution processed, and for me, what makes them interesting is they're super disordered, which is cool. There was a sort of seminal paper from Dane de Quiet, who was also in the University of Washington, I think, at the time, where he published, in my opinion, a quite seminal paper where he measured some scanning electron microscopy, showing the sort of polycrystalline nature of these materials. And on top of it, did some confocal photoluminescence microscopy, and basically found that there were some grains that were like extremely bright, and then by sort of proxy have a high quasi-Fermi level splitting or voltage, and other regions that were extremely dark. And there was a lot to be gained by saying, what if we could turn all of the grains that were dark into the super bright? Like, why are those grains incredibly dark versus other grains incredibly bright? And I think that was a big inspiration for some of my work and the work of many others in the field, for sure. So to sort of tackle this, the first technique that we used is something called hyperspectral microscopy. It's an overly fancy name, in my opinion, but it effectively means that rather than just measuring the intensity of light coming from a particular point in your image, you're measuring a full spectrum. And as we saw kind of earlier, you need the full spectrum if you want to pull out all the information about the quasi-Fermi level splitting and the Urbach energy and things like this. So I don't really want to go into too much detail with this, but effectively the green path is the sort of illumination pathway, and it looks like any sort of conventional wide field microscope where you want to create a nice homogeneous beam of a laser. Then you focus it onto the back focal plane of your objective, and you get a nice big broad illumination spot. The sample will then fluoresce. We'll cut out the laser with some sort of filter, and kind of the magic happens here in this backstage, which is where the light is incident on what's called a Bragg tunable filter. So the filter, it's a bit like a grating, but rather than kind of periodicities in 2D, it's actually periodicity in 3D through like a piece of glass, where the refractive index of the glass has been modulated throughout the thickness of the glass. And it basically allows you to produce a sort of bandpass filter, if you will, with like an extremely small bandwidth. So what happens is the light kind of comes in here, is incident on a mirror. It gets hit onto this sort of grating where a small bandwidth of light is sort of dispersed laterally, kind of according to Bragg's law of diffraction. So you'll have a dispersed set of light kind of emerging here. It'll be bounced around this corner cube. It'll hit back onto the Bragg grating again, where it's collimated. But now you basically have light kind of separated in the X plane as a function of wavelength. And then that light is then focused onto your camera. And so what this means practically for your images is that for a given angle of the Bragg grating, you will basically see the image of your sample, but then superimposed on top of that image, you will have a dispersion of wavelength across the x-axis. And so what you effectively do is you slowly step in small, well-known intervals the angle that the Bragg grating takes with respect to your light, and you will basically sweep out the spectrum of light across your sample. Then if you know that for a given angle of the grating, this is, for example, violet, then you change the angle, this becomes violet, and this becomes violet, etc. You can effectively chop up your data, put it back together, and in the end, you end up with a stack of images. Each one is sort of monochromatic. So you'll have a blue image at whatever, 450, 475, 500, etc. And if you take the intensities of a pixel throughout that stack of images, you'll basically pull out the PL spectrum from that image. And so sort of a random example of some data that you get is something like this, where at every point we have a nice photoluminescence spectrum. There are ways that we need to calibrate this setup to kind of turn it into like an absolute PL rather than a relative intensity. I probably won't go into that in great detail right now, but if anyone has questions about it, I'm happy to discuss it afterwards. And I think one word of caution about this type of approach is because the kind of way it's set up is that the measurement is done from one side to the other. It means that for a given wavelength, the left side of the image, for example, will be taken at an earlier time compared to the right-hand side of the image. And so you need to be sure with your materials that, for example, with the Halide Perovskites, that they are not sort of light soaking very dramatically or phase segregating over the time scale of your measurement. Because if they are, then that will look like a big shift in the intensity and will look like a very large shoulder in your PL spectrum, which is not so good. That's something to be aware of. So this is some data that we took on our microscope, and this is the sort of fit to be able to extract things like the quasi-Fermi level splitting, photoluminescence quantum efficiency, and in this case, the Urbach energy. One finding that we had from some of our perovskites here is that the kind of photoluminescence quantum efficiency, which is sort of this proxy for the voltage, and the Urbach energy, which is this proxy for the kind of below band gap disorder, were actually quite inversely correlated with one another, which kind of intuitively makes sense. The higher the PL, the lower the disorder. And so this is kind of depicted here in this sort of 2D histogram, where you can see there's quite a nice trend. And one thing we found that was interesting here was that the regions that were the highest in photoluminescence quantum efficiency also appeared to be redshifted in their spectrum. And so that kind of got us scratching our heads. We were like, whoa, what's going on? And so we basically want to kind of understand why that's the case. And that's a sort of, I guess, motivation for using a multimodal approach. We see this observation, but without any more information, we're not able to kind of pick apart what is it about that spectrum that makes it that way. And so the second technique we used is hard X-ray microscopy. This is a synchrotron-based technique where you basically take a nice beam of X-rays, and over the course of about 200 meters, you focus the X-rays down to a very fine spot. Using some very clever X-ray optics. X-rays are very hard to focus for anyone who hasn't tried because the refractive index in that wavelength is very close to 1. And so you basically come in at an incredibly grazing incidence into these big parabolic shaped mirrors. And, you know, over tens of meters, you can focus the X-ray down to about 50 nanometers in size. And that basically allows you to, depending on the detector that you use, to create a map of either X-ray diffraction or X-ray fluorescence or both. And so you can kind of produce a map of chemical and structural information about your device or your sample. One thing to note, just sort of as an aside, is that compared to a technique like electron diffraction, X-ray diffraction on the nanoscale is actually very sparse. If anyone can think of like an electron diffraction pattern, there's normally like tens of spots where you can learn a lot about the kind of orientation of a sample. With nano X-ray diffraction, that's not the case, because the Ewald sphere, or basically the momentum of an X-ray photon, is a lot smaller than the momentum of an electron. And so the number of spots that you have available to you to be in the diffraction condition is much smaller compared to an electron diffraction image, but it's still very informative. So with that, we basically can, for example, for every point, have a X-ray fluorescence spectrum. Each peak is sort of characteristic of a different atomic level transition in an element. We can sort of track the variation of these different peaks and produce maps, for example, of the lead content, the bromine content, or ratios thereof. And then similarly, we can also look at variations in the lattice constant from the X-ray diffraction. We can also look at what lattice constant we would expect given the composition of the perovskite from sort of a a Vegard's law type approach, where you're sort of interpolating the lattice constants of, for example, the iodine and bromine rich region. And you can produce sort of nanoscale strain maps. And so this is an example here. The kind of black regions are the areas where there's no data because there was no diffraction in that spot. So all that being said, we then, okay, I caution myself here. This technique does not make perovskites happy. Let's put it that way. If you blast a very high energy, high intensity X-ray beam at perovskite, it will not thank you very kindly for it. We've done quite a bit of work, and some colleagues in the group have done quite a bit of work on studying beam damage, and it's very real in this technique. And so if you plan to do sort of a multimodal set of experiments, and this is one of them, this should be the last one, because you shouldn't be measuring this device afterwards. That has to be said. So we point our hyperspectral and X-ray microscope at the same spot. The first thing that we did was we looked at, does strain, microscopic strain, affect the disorder or the photoluminescence quantum efficiency? And the answer, kind of interestingly, is not really. These are basically correlations between the strain and the Urbach energy for different peaks. This is the 100, 200, and 210. If you kind of run the statistic numbers on this, there's effectively no correlation whatsoever. And so we kind of thought that was quite interesting, that strain microscopically doesn't appear to play much of a role, at least in our samples. So we looked at the chemistry next. Here's, again, an example. This is a ratio of bromine to lead, which I'll refer to quite a bit. And when we sort of look at the areas that are the most bromine rich, and we plot them over in blue of an Urbach energy map versus the areas that are the most bromine poor, now we're starting to see something, which is good. We're looking at a lot of different samples, a lot of different maps, and convincing myself. We basically found that the regions that are the highest in bromine are the lowest in disorder and the highest in photoluminescence quantum efficiency. And so that's kind of schematically shown here, where the blue regions have a very statistically significantly lower Urbach energy, higher photoluminescence quantum efficiency. And so we kind of get this notion of the high bromine to lead ratio being somehow better. But that was sort of a bit of a paradox to us, in the sense that in the previous slides, I told you that the best regions were kind of a lower energy emission, a redshifted emission. And for those of us in the literature, we know that more bromine should mean a blue shifted emission. And so we needed to figure out what was going on there. So I said there. So we turned to a technique which I am not an expert on, but some very smart people in the FAST lab in Cambridge are. This is transient absorption microscopy. It's basically the equivalent of transient absorption, where you kind of pump in an excited population of carriers and then probe that population of carriers at some finite time after the pump. And you can basically track the charge carriers as a function of time and energy. But then additionally, in a microscope, we can basically track them as a function of space too. And so in the interest of time, I'll move through this quite quickly, but we basically now do sort of a hero experiment that I wouldn't necessarily recommend anyone does, where we pointed photoluminescence, X-ray fluorescence, and transient absorption microscopy on the same spot to try to figure out what was going on. Due to the geometry of the transient absorption, we had to use a line cut rather than a full image. We had to give up one of the spatial dimensions for an energetic dimension. On this line cut, the black line is the bromine content. The red line is the PL of the same spot. And so you can see in a lot of places, they kind of correlate with each other quite well. When we look at the transient absorption data, this is basically the ground state bleach energy. So it's kind of the average energy of the excited state carriers. This is, again, as a function of position. The y-axis is a function of time, and then the color scale is the energy. So dark energies or dark colors are high energy. We see that for the bromine to lead rich areas, they start at high energy, which we would expect, and the bromine poor areas start at low energy, which we would expect. But very interestingly, in the bromine rich area, over the course of a few hundred picoseconds, the carriers are found to kind of very dramatically redshift, while the bromine poor regions show kind of no such redshift. And that's kind of shown here, where we have kind of a shift in the ground state bleach to the left, whereas it's just downwards here in the ground or the bromine poor regions. And so that led us to produce sort of an understanding of what's going on, in that it seems that in a lot of the bromine to lead rich regions, they actually have small low energy inclusions in them, which are iodine rich. And that sets up sort of an energetic gradient where carriers can kind of funnel down into. It increases their local carrier concentration and kind of pulls them away from trap states and allows them to kind of radiatively recombine more effectively. And so that was quite interesting. So that's kind of what I've said here. What about full devices? I think I'm kind of coming quite close to the end, so I might rush through a few quick takeaways here. Contacts in a full device also add additional non-radiative recombination pathways. This is sort of a plot of showing that the quasi-Fermi level splitting of a bare film is much higher than lots of different contact materials. So if we want to understand the full device in terms of the voltage, we also need to include contacts. Contacts can also act as substrates, which can change properties. And of course, the critical one, which is that contacts also need to transport charges. And so all of the previous stuff I've shown you is only talking about the voltage. And in principle, a solar cell is not just its voltage. It's much more than that, you know. So we want to probe the whole JV curve. We get this, and then sometimes in the literature, we occasionally get this with things like laser beam induced current, but we really want the whole thing. And so that's where some people in the literature kind of come up with voltage-dependent photoluminescence. This is where we effectively scan the voltage on our solar cell and measure the photoluminescence as a function of that voltage. Martin Stolterfoot and co-workers, they basically found that there was a very tight correlation between the actual electrically measured current versus voltage curve and the PL as a function of voltage. And I maybe won't formalize it right now, but on the next slide, I can show you there's some mathematics which suggests that if you basically take the difference in PL between open circuit and any finite voltage, normalized by the PL at open circuit, this is actually a proxy for the electrically measured current as a function, or normalized by the generated current. And so a couple of clever folks in the literature, they basically said, well, if we could do this in the bulk, could we not do this in the microscope as well? And so we could, in principle, reconstruct current versus voltage curves microscopically. I'll skip this for now in the interest of time, but there's some maths. Makes me credible, something physicist. So with that, we can basically take our nice solar cell and we can measure the photoluminescence as a function of voltage. And you can see that for this particular device, at open circuit, it's super bright. And as soon as you start to extract carriers, there's less carriers there to recombine. And so the PL sort of decreases. And from that, you can sort of reconstruct JV curves as a function of space, which is really cool. We did some drift diffusion simulations, not as fancy as the ones that Vincent does for sure, but we basically wanted to see, you know, in sort of realistic scenarios and not just in the case of an ideal diode, what are we sensitive to? And in short, we're sensitive to quite a lot with this technique. Series resistance, these sort of dotted lines are the PL versus voltage curves, and the solid lines are the actual current versus voltage curves. So they're totally overlaid. With shunt resistance, we actually miss this because some current kind of flows parallel to the diode. And so that is a thing where if you're using this technique and your devices are heavily shunted, you might think that they're better than they actually are. If you have lots of mobility problems, you can still detect that very nicely. If you have extremely short non-radiative lifetimes, you will overestimate your fill factor with the PL. If you have some surface non-radiative recombination, you actually don't really see much of a change. And if you sort of plug in a bunch of parameters relevant for our own personal devices, you can see there's actually quite a nice match between one and the other. So I realize I'm running over time. I'll maybe zip through this really quick. We basically point all the things together, X-rays, and operando microscopy, and we can produce these incredibly rich data sets where on a given spot, we can look at the photoluminescence spectrum, we can look at the composition, and we can also look at the current versus voltage curves, which basically allows you to say on the same point, what is the quasi-Fermi level splitting doing? What is the chemical composition doing? What is the short circuit current and the optical PCE? We can, for example, show you that operando gives you some insights sort of beyond VOC. This is a particular device where we stress tested it for an extended period of time. And if you were just to look at sort of the optical microscopy at open circuit, this is the quasi-Fermi level splitting before and after, you would actually say like, oh, wow, my solar cell has actually improved over time. I'm a genius, PC improving over time. And similarly, if you measure the XRD, you see no change in the bulk diffraction of the perovskite. So again, suggesting everything is all good. Whereas here, this is a sort of map of the PCE, current versus voltage curves, and PL spectra extracted from a few different points. We basically see that beforehand, it's quite homogeneous. Afterwards, again, as we said over there, the PL is pretty homogeneous again. But we see kind of, you know, I've kind of given away the surprise by like explaining it in this way, but we see really big differences in the current versus voltage curves microscopically. In particular, we see really large changes in the local hysteresis of this device. And we think this is attributed to like a big edge effect where you have lots of mobile ion concentrations moving in from the edge of the device. And so this is just kind of a motivation for why sometimes we might want to characterize our devices beyond open circuit. I will probably skip over some more slides in the interest of time, but we can talk about it more. In a nutshell, we kind of developed these sort of microscopic proxies for performance and stability in these devices. We basically found that if your kind of disorder in your power conversion efficiency microscopically is lower, then your overall performance will be higher, which is maybe an obvious point. We basically found for lots of different types of devices, lots of different architectures and perovskites, that this seems to kind of generally hold as true. So basically, like, the perovskite can actually tolerate quite high chemical disorder and band gap disorder, but really it seems that charge extraction disorder, it's not tolerated very well. We basically found that you can kind of tune this PCE disorder with composition quite easily. And we actually see a nice trend where lower PCE disorder actually corresponds to lower loss of performance over time. However, we basically found that for modifying the interfaces, this can actually be quite a double-edged sword where for some passivation treatments, you can improve the PCE disorder at time equals 0 quite a lot, but then the devices sort of blow up before your very eyes. And we do not want solar cells that blow up. So as sort of a summary, nice picture, multimodal microscopy is sort of a powerful approach to understand the complex behavior of complex devices like the ones that we all work on. We kind of need to take into account limitations and caveats of each technique if we're able to kind of pull out the most information. Carrier funneling can be important in some mixed halide devices. And the operando microscopy reveals sort of that charge extraction is really important to sort of homogenize across your device. So I'd like to acknowledge some great collaborators and teammates. I've been working a lot with Cullen Chosey recently. He'll probably be on the job market in the next year or so. So if you're looking for postdocs or whatever, you should look him up. Some great collaborators in Diamond and in Helmholtz, as well as the Strengths Lab. So with that, thanks very much for your attention.